Today, an FDA approval in lung cancer, promising findings in Hodgkin lymphoma, a complete response letter issued for a biosimilar, exciting results in multiple myeloma from the 22nd Annual European Hematology Association Congress, and a recommendation for a kidney cancer approval. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved the combination of dabrafenib and trametinib for patients with BRAF V600 positive advanced or metastatic non-small cell lung cancer. In the phase 2 trial that led to the approval, the overall response rate was 61.1% in patients who were treatment naive, and 68% of patients did not show progression at a median follow-up of 9 months. This cohort had not reached endpoints for median duration of response or progression-free survival at the time of approval. However, among patients who responded to treatment, 59% had responses lasting longer than 6 months. In patients who were previously treated, the ORR was 63%. Researchers found that the response was durable with a median duration of response of 12.6 months. The FDA also approved the Oncomine DX Target Test, a next-generation sequencing test that detects the presence of BRAF, ROS1, and EGFR gene mutations. This is the first NGS oncology panel test the FDA has approved for multiple companion diagnostic indications. The European Union approved the dabrafenib-trametinib combination in April 2017. In classical Hodgkin lymphoma, phase 3 findings showed that frontline brentuximab vedotin was associated with improved progression-free survival compared with standard chemotherapy in patients with advanced disease. Results of the global multi-center Echelon-1 trial demonstrated that the 2-year modified PFS rate for brentuximab vedotin plus adriamycin, vinblastine, and dacarbazine was 82.1% versus 77.2% for patients receiving adriamycin, bleomycin, vinblastine, and dacarbazine. The brentuximab vedotin combination reduced the risk of disease progression or death by 23%. An interim analysis of overall survival also showed a trend favoring the brentuximab arm. More data will be presented this December at the 2017 ASH annual meeting. Echelon-1 enrolled 1,334 patients with a histologically confirmed diagnosis of stage 3 or 4 classical Hodgkin's lymphoma who had not been previously treated with systemic chemotherapy or radiotherapy. Study sites included medical facilities on every continent except Antarctica. The FDA has rejected a Biologics license application for the epoietin alpha biosimilar epoietin Hospira. The agency cited manufacturing concerns at the company's fill-finish facility in McPherson, Kansas. The decision marks an uncommon situation of the FDA denying a drug recommended by the Oncologic Drugs Advisory Committee, which voted 14-1 to approve the BLA on May 25, 2017. Pfizer, the manufacturer of the biosimilar, received a warning letter from the FDA in February 2017 detailing 5 areas of concern, including in-process specifications, failure to follow procedures to prevent microbiological contamination of drug products, and a lack of scientifically sound and appropriate sampling plans for inspection and analytical activities. Moreover, the FDA argued that Pfizer should have known the McPherson site might have problems since the FDA cited 5 other Hospira plants for similar problems from 2010 to 2015. Pfizer stressed that none of those issues specifically relate to epoietin Hospira, though the McPherson plant was a potential manufacturing site. Additionally, the FDA did not request more data to support a further approval as it did in 2015, when the agency rejected an abbreviated BLA. Pfizer said it submitted a corrective and preventative action plan to the FDA in March and is working to address the concerns listed in the warning letter. Hospira was seeking approval to treat anemia due to chronic kidney disease, to treat anemia in HIV-infected patients being treated with zivudidine, to treat chronic renal failure, and to reduce the need for allogeneic red blood cell transfusions in patients with perioperative hemoglobin from more than 10 GDL to less than 13 GDL who are at high risk for perioperative blood loss from elective non-cardiac, non-vascular surgery. Phase 1 data presented at the 22nd Annual European Hematology Association Congress demonstrated that patients with multiple myeloma achieved a response following treatment with an active dose of BB2121, an investigational anti-B-cell maturation antigen chimeric antigen receptor T-cell construct. The objective response rate of the CRB 401 trial was 100% in 9 evaluable patients who had been infused with BB2121 at a dose of 5 × 10⁷ CAR-positive T cells or higher. 27% of patients achieved complete response, and 75% of patients demonstrated a very good partial response or better. The evaluable patients at this dose level were minimal residual disease negative. Furthermore, the ORR across all dose levels was 89%. The median time to first response was 31 days, and the median time to best response was 50.5 days. The median duration of response was 134 days. The ongoing study is enrolling at 7 sites to an anticipated total enrollment of 50 patients. The European Medicines Agency's Committee for Medicinal Products for Human Use granted a positive opinion recommending marketing authorization of Teuvos for adult patients with advanced renal cell carcinoma. The opinion was based on results from the Phase 3 Teuvos trial, which evaluated the efficacy and tolerability of Teuvos compared with sorafenib. The median progression-free survival in the Teuvos arm was 11.9 months compared with 9.1 months in the sorafenib arm. Additionally, treatment-naive patients who received Teuvos enjoyed a greater improvement in PFS. For those patients, the median PFS was 12.7 months versus 9.1 months in the sorafenib treatment-naive arm. At the time of the overall survival analysis, mortality rates were 45.4% in the Teuvos group and 39.3% in the sorafenib group, corresponding with a stratified hazard ratio of 1.245, trending in favor of sorafenib. The median OS was 28.8 months in the Teuvos arm and 29.3 months in the sorafenib arm. Should the full committee accept the positive opinion, Teuvos would be approved for use in the 28 European Union member states, plus Norway and Iceland, to treat adults with advanced RCC who are VEGF-R and mTOR pathway inhibitor naive and are either untreated or have failed prior therapy with interferon alpha or interleukin-2. This week, Dr. Richard Boulay of Lehigh Valley Health Network discussed recent advancements in gynecologic malignancies.
So we are involved in a number of clinical trials. Recently, we've also formed an institute with with Memorial Sloan Kettering, their alliance, and we're working together with them to do a couple surgical trials. We're doing one where we're adding a device called a wound vac over people's closed wounds to minimize the likelihood that that wound will open up and cause delays in subsequent chemotherapy and quality of life issues. We have diseases, uterine cancer, where we're adding radiation to chemotherapy and see if one is better than the other, or there are combination of the two is better. So there's a number of clinical trials, plus, as I said, starting to add these biological molecules. We've worked a lot in the past year with some of the immuno-oncology drugs. The trouble is they are, when we're looking for things like ovary or cervix cancer, we're really only looking at 80 folks nationwide. So they're quick trials, and we're waiting for some of that data to come back now to see how effective they are and if it's worth putting them in the armamentarium of treatment measures for these diseases and where they should be in the next phase of clinical trials. Do we combine them? Do we use an either or? So there's a number of brighter minds than mine that'll be working together to find out where's the best place to utilize these newer tools.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.